People are joining, so I'm taking 5 more minutes because today is my first class. I want maximum people to join. So just connect with me. Okay. Good evening, good afternoon, everyone. I am Shikha Kaushik. I will be taking your GATE coaching classes on every Saturday, 3 to 4 p.m. And to get a good results and to maintain the discipline throughout the course, I would request you to stay connected every Saturday because that's how we're going to progress towards our GATE preparation, and that will definitely going to make the session more appropriate for yourself. So so before starting with my subject today, I want to know how you're going to go through, how do you want me to take you through the subjects? Because today we have just 1 hour class. We always going to have a 1 hour class, and I don't want to miss previous year questions. So I have prepared, my course like that I'll take you through the questions, and then we'll discuss whatever theoretical aspect of those questions are, or the practical aspect of those questions are. So take you through the question, and then we'll discuss about various concepts. Is it fine, or you want me to give you some content before taking questions? So I would encourage you to write in comment box, or you can turn off your mic and tell me whatever way you want. I'll take my coming session like that. So today I'll be taking you through the questions, but if you think that you want some concept before taking any previous year question, I can do that too. So if you have any suggestion, you can turn on your mic and tell me whatever you want, whatever you expect me to do during our sessions.
So, ma'am, you can go with the questions, and if necessary, then you can explain the theoretical
Yeah,
portion
yeah,
of the
I
questions.
am. Hmm.
And in some cases where theory is mandatory before explaining the questions, then you can do that in that format also.
Okay. Shreyansh, can you just, I think somehow you have pinned to my screen and I'm unable to open it. bacteria. Okay, fine. So thank you for your suggestion, Shreyansh. I will definitely planning to do like this only. I'll take questions and side by side I'll explain the theory related to it. So to begin with, today we are going to discuss cell biology. Cell biology is one of the very important part in our GATE biotechnology paper. You will definitely going to have two or three questions from this subject, and I'm going to take question from 2011 and try to complete as much as I can. So begin with cell biology. Okay, fine. Before telling you what all I'm going to teach, so mostly my session will be divided into four parts, where I'm going to talk about give you a brief overview of what all we have to cover in a particular subject, and then I'll definitely going to share some tricks and tips so that you can remember those points and they will help you to crack the paper. And definitely we'll go through previous year question paper and we'll discuss answer with explanation. And at the last you can give me you can ask me questions, or you can give your suggestion, or you, if you want that some concept needs to be taken in more details, so we can go through that as well. So if you talk about cell biology, which come in section 3 of our GATE biotechnology exam, and we have these few topics which are given as a main topic for the paper. First is prokaryotic and eukaryotic cell structure, where we have to you have to learn about bacteria as well as eukaryotic cell and all the organelles that are there in the eukaryotic cells. Then we have cell cycle and cell growth signaling. You need to read this topic in detail, because there are going to be more questions from this section, especially from the cell cycle and all the CDPKs and other cyclines that are responsible for cell cycling. All these type of questions will be there from this section. And coming to the cell-cell communication, they're going to be a lot of, we have different cell signaling. We have different type of, cell signaling molecule. We have junctions. We have our pathways. So all these things need to be done in this particular section. Then we have cell signaling and signal transduction, which will talk about all the signaling that happens whenever we have a ligand or hormone. These things has to be done in this particular section. And post-translation modification and protein trafficking involves Golgi, ER, and there are some hot points from this particular subject, this particular section. So we'll discuss that also. And then we have cell death and autophagy, which is another important topic for GATE. And we will have few tricks which will help us clear this particular question from this particular section. So we can able to do a question very quickly if we remember those tricks. Then okay, fine. So coming to the prokaryotic cell. So this is just a overview. I'm not going to get into detail of the concept. This is a bacterial cell. There you have different type of organelles. You have to remember each and every organelle and their respective function. Likewise, we have one of the very important aspect that you need to remember is difference between gram positive versus gram negative. Apart from not just the orientation of cellular membrane, but you have to remember that what is present in gram positive, that is teichoic acid, and what is not present in the gram negative, which is not present in gram negative bacteria. So in gram negative bacteria, the membrane organization is given like that we have three different type of protein, which is antigen, then we have a core protein, and then we have lipid A. This part of the gram negative bacteria is very important. You will get many questions from this particular area, that whether lipid A interact with core portion of the membrane, or it interact with the outer membrane, inner membrane. Such kind of question you will face. You will definitely going to see if you're going to do previous year questions. So this particular part is very important. That's why I have noted it down for you guys. Next is animal cell culture. In animal cell culture, you have to go through each each and every organelle, you have to remember, you just need to point out few things. You just remember a few things first, the organization of that particular organelle, whether it's a single membrane or double membrane, or the function of that particular membrane, especially when you have endoplasmic reticulum and Golgi body, where you have there is smooth ER and rough ER. In that, you have sometimes you get questions which are very confusing, which make you mark the question wrongly. So you have to remember that what smooth ER is doing and what rough ER is doing, and how they are associated with Golgi. It's very important in terms of gate examination. So coming to the first question is that is there on your screen. The question says that the liposaccharide present in a bacterial cell wall has a lipid A which is connected to first O polysaccharide, core polysaccharide, both O and core polysaccharide, or ramenose mannose disaccharide. Can any of you answer this question? Are you aware of this particular aspect? If anyone have the answer, even if you don't, let me take you to the answer of this particular question. So as I mentioned, the organization of plasma membranes in animal culture as well as in plant culture and bacterial cell is very important. This is a very important topic of cell biology, and you will definitely going to get one or two questions from this particular part. So lipopolysaccharide in bacterial cell is basically contain three part. One is lipid A, which I already shown you in the figure. I'll show you again. Then you have a core oligosaccharide, and then O chain. So the lipid A is placed inside in the inner side of the cell, towards the inner membrane. Then we have in middle, we have a core oligosaccharide, and the O chain is outside. So generally the question asked whether lipid A interact with is connected with O polysaccharide, core polysaccharide, or both. So if can any of you now answer this question, because I have given you the three part. Lipid A is inside. Okay, so if you could see, so lipid A is inside, then we have core polysaccharide, and then we have O polysaccharide. So if you could see that good Shreyansh, if you could see A is only lipid A is only connected to the, if you could see here, lipid A is only connected to the core polysaccharide. So the answer for this question will be B. That is, the lipid A is only connected to the core polysaccharide. I hope you all got the answer. If you have any doubt, please let me know. Otherwise, we'll move to the next question. So likewise, you have to remember, just remember that you have to understand plasma membrane, be it bacteria, be it eukaryotics, plant, animal, both, because you will definitely going to get some question related to plasma membrane. Okay, so the organization of plasma membrane is very important. Okay, shall I move to next question? So the answer for this is core polysaccharide. Uh, I would request you all to be a little interactive with this session, because I'm doing this just for you guys. I want you guys to interact with me. That would make your learning much more, effective. So even through the comment section, you can interact me through the comment section if you want, but do interact me with the during the session. That will make this learning more, fun as well. Okay, coming to the, okay, I have also given, so there will be, you might be, I mean, I don't know, NPTEL will share this PPT with you or not, but I will provide you as much, as much resources as possible. So I have given a link where you can read about lipopolysaccharide and lipid A, and all question will come from that particular, I mean, if you read that particular section, you will be able to do all the questions. So coming to the next question, where you have a question which says, it is a GATE 2012 question, choose the correct signaling transduction pathway. So if you're going to study signal transduction, where you have a particular hormone that binds to a receptor and it activates a downstream molecule which leads to activation of certain expression of certain genes. So this is in the section of cell signaling pathway. So you have four options where there is hormone which interact with, first option say hormone which interact with 7-TM receptor, then it binds to G protein, it then activates CAMP and then PKA. Likewise, we have B, C, and D. If you have answer for this question, let me know. Otherwise, I'll take you through the this thing only. So in G protein signaling, you have a hormone. That hormone binds to this hormone actually. Wait a minute. Okay. So the hormone binds to G-coupled hormone receptor, which you can say 7-TM receptor. It is a transmembrane receptor which has 7 subunit. That's why it's called 7-TM receptor. It binds to a hormone binds to that and it leads to activation of G protein. Once the G protein activated, there is a phosphorylation that leads to GTP and G protein release, and it binds to adenyl cyclase. Once the alpha subunit of G protein binds to adenyl cyclase, it activates the formation of CAMP. Once CAMP is produced, it activates protein further PKA, which leads to activation of many genes. So this is the correct sequence. So if by this sequence we go, we will have the answer as A, where hormone is binding to 7 transmembrane receptor, which is a G protein coupled hormone receptor, and then it leads to the phosphorylation, and that phosphorylation leads to the release of G protein, which is an inactivate state. The alpha subunit of that particular protein binds to adenyl cyclase, which leads to production of CAMP, and that further activate PKA. So this is the entire sequence. So the answer for this question is answer A. Okay, so if you're going to remember all these sequence, whenever you do signaling or signal transduction, or if you read any pathway, just try to make your just try to write down the steps of that like it's written here. So that will make you remember everything. So remember one thing, that hormone always going to bind to a receptor. Once it bind to a receptor, because it's a G protein signaling pathway, it will lead to activation of G protein, and then there will be a downstream activation. So by that logic also you can do this question. So you second all the other option, especially the option B, which say hormone bind to directly to G protein. Hormone cannot bind to, or any ligand cannot bind to a protein directly. It will definitely going to bind to a receptor to make a particular pathway active. I hope I'm not moving too fast. If you have any doubt, please raise your hand or comment in the chat box so that I can take your questions. Coming to the next question, which is again a GATE 2012 question. You have different chemical compound or enzymes, and then sorry, there's a set of chemical compound, and there are group 2 where you have particular enzyme they will act on. So starting from the MTT, so you have to match the following and you have to come to the answer. So the first compound here is, first chemical salt here is MTT, which we use to test the cell viability. So it is a tetrazolium salt that is commonly used to detect reductive metabolism in cell for viability, proliferation and cytotoxicity assay. So how it works, MTT actually is of a yellow color. It's a tetrazolium salt which is of yellow color. It has an ability to penetrate in the cells. It go inside the cell and it interact with mitochondria. There in mitochondria we have succinate dehydrogenase which reduce it to form formazine. So it turn into formazine, and this formazine is dissolved in DMSO, which once dissolved in DMSO give purple color. So that's how we do our cytotoxicity or cell viability assay. If our cells are viable, they will not have a leaky mitochondrial membrane. And if we have there is a cytotoxicity present, then our mitochondrial membrane will be leaky. So the enzyme will come out, and this enzyme will turn MTT into formazine. So more the yellow color, more the purple color we get, more cytotoxic effect is there. So MTT is for succinate dehydrogenase. So for MTT we have P is 2. I hope you are fine. I hope I'm going in a perfect speed. Please let me know if I'm too fast. I can slow it down. Okay, Ram have mentioned that would question-wise teaching cover all concept? It might not cover all concept, but at least you will have an idea what all topic you have to cover, because mostly question repeats in GATE exam. So if, for example, if I'm talking about MTT, you might not get a direct question like this, but you may get whether MTT is used for cytotoxicity or any other process like that. So I will try to take concepts as well. If you have doubt in any particular concept, I will take that. But before, because we have a very limited time left, and I will have 1 week, one class per week, so I don't want to miss any topic, so that you can cover maximum part. And if you have any doubt, stay connected with me. I will provide you concept wise resources as well, or we can discuss a particular topic here in the class as well. So today is the first class, so I thought that it would be better that if I can um take the session question wise only. Okay, so coming back to the question, and for MTT, it's succinate dehydrogenase, which is present in mitochondria. Again, I'm telling, if the cell is if the cell has a cytotoxic environment, the mitochondria membrane become leaky, and the succinate dehydrogenase come into cytosol, and due to I mean this enzyme, the MTT convert into formazine. So more the purple color, more the cytotoxicity. Fine. So next is annexin. So annexin 5 is a cellular protein of the annexin group, and it has affinity toward phosphatidylserine. So it binds to phosphatidylserine. So this ability of annexin 5 is being used in flow cytometry. So if there is a cell death. Okay, so whenever there is an apoptosis, so what happened is there is a loss of plasma membrane organization. So what happened when there is a cell death, plasma membrane, the way plasma membrane is organized is not present. I mean, the cell will lose its organization. The moment cell lose the cellular organization, it will lead to the phosphatidylserine, which is placed in the inner membrane, will come at the outer membrane. The moment it will come outer membrane, annexin 5 bind to it, and that's how we detect whether there is an apoptosis or not. So annexin 5, it has a ability to bind to phosphatidylserine. So if you see the question, so we have annexin 5, and there is a fourth option which says phosphatidylserine. So annexin 5 match with the phosphatidylserine. Coming to the next, we have methotrexate, which is an anti-metabolite, which is mostly used in chemotherapy, and it is also an immunosuppressant. This drug is used to treat cancer, to treat autoimmune disease like cirrhosis, and it is also used in arthritis. So what is the mode of action it is? So that's how we get to know whether it's bind to which enzyme. So if this drug binds to which enzyme. So what happened? So MTX has an affinity to bind to folic acid. So but it do. I mean, it basically block the iron. It basically blocked the DNA synthesis. How? By actually interfering in the formation of purine synthesis. During the purine synthesis, and it interfere and bind to inhibit an enzyme which is very important in purine synthesis. So what it does, it binds to dehydroxy um, okay, it's dehydroxy. I think I've written something wrong here. Okay, yeah, it's dehydroxyfluoro. It's a floxate folate reductase. So it is basically it leads to the degradation of folic acid and removal of iron from there. So this leads to formation of thymodylate. Thymodylate is used in purine synthesis. So if this if MTX bind to DHFA, which is a reductase enzyme important in purine synthesis, it will lead to less purine. I mean, purine synthesis will definitely block here, and we will just have pyrimidine. And if we just have pyrimidine, we cannot further move for cell. There will be no. There will be no DNA synthesis. So if there is no DNA synthesis, cell will leave. There will be cell death. So this property of MTX is used, and this drug is used in cancer as well as many autoimmune disease. So for methotrexate, we have the dihydrofolate reductase. So R goes to 1. Okay, I have given more details about this thing. So if you want to read, you can read it here. I think PPT will be shared with you, so you can read this thing and you will keep it with you whenever you want to revise. Next is Taxol. Taxol is a very commonly used, drug during cancer treatment. It is also called paclitaxel, and it is mostly a natural source this has been originated. There is a origin of Taxol, how it has been done. But just to tell you, mostly it is mostly used in cancer, especially in breast and lung cancer, especially when they leads to metastatic stage. What it does, it leads to depolymerization of our tubulins. So once there is a depolymerization, there will be no cell cycle, because we need tubulins. We need our cytoskeletal membrane, our cytoskeletal structures to do for cell to proceed for cell cycle. So for Taxol, which is also called paclitaxel, is basically microtubules, because it leads to depolymerization of microtubule, and there will be no cell cycle. So I'm also giving you the effect of these drugs or these chemicals, but at the same time their affinity is towards these enzyme or this particular structural organelle. So if we match all of this, we will have our answer as, okay, it will be, for MTT, it is succinate, which is P2. So once you have answer for one, which is MTT, you will able to eliminate rest of the option. So A and D is eliminated. B and C, it's B only. So for Q, we have annexin 5 bind to PS, which is phosphatidylserine, and then we have MTX, which bind to DHFR, and Taxol, which binds to microtubules. So answer is B. Okay, next is cholera toxin, and the question says that cholera toxin increases CAMP level by modifying which of the part of G protein. So if I take you through the signaling of cholera toxin, what happened? Cholera toxin is also contain a pentameric B subunit, which is exactly like the B subunit of our G protein signaling, G protein signaling pathway. So what it does, the B tetra, if you could see here, this pentameric B subunit binds to GS alpha. So remember this thing, that it binds to, it actually take a place of AB. It actually replaces the B subunit of G protein, and it binds to GS alpha, and that's how we can solve a question, because it say whether it's modified GI protein, whether it's modified GS protein, whether it bind to a adenylate cyclase directly, or whether it activates CAMP phosphodiesterase. So basically, it directly bind to modifying GS protein. Once it bind to GS protein, what happened? When the GS alpha subunit is bound to adenyl cyclate, which is here, it leads to formation of CAMP. So once the cholera toxin infect a particular cell, I mean a particular organism, because it leads to, I mean, it leads to the, it may leads to basically it happens through water, and it can lead to a death of a particular uh, organism as well, because our cells start to reject water. So we will not have any water inside us, because our sodium chloride ions will, I mean, our transport, a sodium potassium channel will remain open, and there will be uh, uh, there will be water will remove out of the cell. So body will not retain water, and that can lead to the death of an organism. So what happened is that it bound to GS alpha, and it leads to continuous supply of CAMP, which leads to opening of this particular channel, which is sodium chloride channel, and leads to removal of water. So if you have to solve this question, it just bind to GS alpha. It is not binding to GI. So the answer of this particular question is B, that in chlorotoxin CMP level increasing, because जो chlora cholera toxin है हमारा, that has a beta subunit which is actually exactly replicating the beta subunit of G protein, and it binds to GS alpha and leads to it actually lead a bound form of alpha subunit, because now the alpha subunit of G protein will not bind to this beta subunit, and this released alpha subunit bind to adenyl cyclate, and that will uh, supply content, that will increase the production of CAM, and that leads to the death of a particular organism, because it leads to opening of water channel. All the water pump will be open, and the water body will release all the water. It will not let water retain inside the body. Okay, so just to know whether what is our normal G protein pathway. This is the normal G protein pathway, which I have given for your reference. So here, if you can see that if G protein is bound to adenyl cyclates, it's lead to CAMP production, because the beta subunit is not bound to alpha. Once this alpha subunit of G protein release, it will lead to formation of CAMP. So to maintain this thing, generally this beta subunit remain bound to alpha subunit, and the G pathway remain inactive. Once alpha subunit is released, it remain active. So that is what cholera toxin do. It lead to release of alpha subunit, and that leads to increase of CAMP. So this I have given just for your reference, that how G signaling pathway work. Next question is the cytokinetic organelle. Cytokinetic means division of cell. Okay, when there is a cytokinesis happen. So cytokinetic organelle is which organelle that lead to cytokinesis in plant cell. So there are four options: centroid, phragmoplast, proplastid, or chromoplastid. So which of this organelle is involved in cytokinesis? So if I talk about cytokinesis is the physical process of cell division which divides the cytoplasm of the parental cell into two daughter cells. So when there is a physical division of a cell, that is called cytokinesis. So if there is just a nuclear division, what it is called? Can anyone answer that? If there is just a nuclear division, it is called it is called karyokinesis. So if there is a
Karyokinesis.
cell, sorry,
Yeah, that is karyokinesis.
Yeah, so if our nucleus divide, it's called karyokinesis. It happens when there is no physical division of cell, but once the physical division of cell happen, then only we have two daughter cells with us. So cytokinesis is basically a physical division of cell. Okay, but and what is plant cytokinesis? Plant cytokinesis is involves the formation and insertion of new cell wall. So basically, plants have a very rigid cell wall. You know that, right? Plant contains cell wall. So when there is a division of cell wall, when there is a division of uh, when there is a formation and insertion of new cell wall which leads to formation of two daughter cell, that is plant cytokinesis, and it happens at the phragmoplast. So what exactly phragmoplast do? Phragmoplast is basically help in organization of cell wall, and that leads to, so here we have phragmoplast, this come together and organized a new cell wall here, and that leads to the division of plant cell. So Phragmoplast is responsible for the final stage of cytokinesis in plant. So just remember this thing, that cytokinesis happen in plants through phragmoplast. So the answer is B. I have also given you how cytokinesis happen in animal as well as in plants. So in animal there is a formation of furrow, which is a cleavage furrow, which leads to a division of two cells, and it happens during the cell cycle only. Likewise, we have things in plant cell. So I have given you to compare both the processes. I have given that for you so that you can refer it later. Next question is, which one of the following is incorrect about a typical apoptotic cell? So as you know, apoptosis is a programmed cell death. It leads to a cell death of a particular cell in a very programmed manner. Okay, so first option is phosphatidylserine is presented on the outer cell surface. Cytochrome C is released from the mitochondria. Mitochondrial membrane potential does not change, and Annexin 5 binds to the cell surface. So, can anyone answer this question? Because we have talked a little bit about apoptosis B when we are talking about Annexin 5. So, this will help you to solve this question. If you can recall that, because I have mentioned phosphatidylserine there, I have mentioned cell organization there, cellular organization which we lose during apoptosis. So जब हमारा cell apoptosis के through process करता है, मतलब when it have a programmed cell death, it loses its cellular organization. So that leads to the movement of phosphatidylserine to the outer cell membrane, and that's how annexin bind to it. So दो option तो for sure correct है इसमें. Rest two, I have given you a hint as well. Rest two, you have to choose between B and C, which one is incorrect. Okay, fine. I'll take you through the options. First one is phosphatidylserine is presented on the outer cell membrane. So what happen whenever there is a cell death? क्या होगा cell death के अंदर? कि हमारा जब cell die हो रहा है तो उसकी जो overall organization है, it will lose. So it will have a disintegrated DNA. DNA will be fragmented. Cellular organization will not be there. Cell will dismantle. Basically, सारा cell dismantle हो जाएगा. The moment it will get the cellular organization loses, phosphatidylserine, which is first option, is correct, and the phosphatidylserine will come to the outer cell surface. So first statement is correct. Next one is cytochrome C is released from the mitochondria. If you guys have studied about the apoptosis as I mentioned is a programmed cell death. There is a release of cytochrome C from mitochondria. Once the cytochrome C released, the mitochondria become a more acidic. So its membrane potential also change. So if you see cytochrome, second statement is correct, that cytochrome C is released from the mitochondria, and once this cytochrome C release, the membrane potential of mitochondria change, the mitochondria become more acidic in nature, and obviously annexin 5 binds to the cell surface when we have phosphatidylserine. So here in the statement C, it is written mitochondrial membrane potential does not change. It does change because there is a loss of of cytochrome C from the mitochondria, and that leads to, because cytochrome C is involved in the movement of iron. Okay, and once the molecule which is responsible for the movement of ions, that is mostly hydroxyl ion or H plus ions. So if the if the molecule which is responsible to maintain the mitochondrial membrane potential is released outside the mitochondria, it will definitely lead to the change of its potential, because now there is no balance which is being maintained. So C option is incorrect. So your answer is C, that mitochondrial membrane potential does not change. It does change during apoptosis. Rest all statement is right. I hope you are able to relate to NXN 5, phosphatidylserine, cytochrome C. I hope you will grasp from here that what all points are important whenever we talk about apoptosis. So you may get questions some or other way around these particular points only. So try to remember as much as possible. Try to remember important point, and you will be able to solve the question in your coming gate exam. Okay, so just for your knowledge, I have divided apoptosis. I have given you a one important aspect of cell death. One is the direct cell death, which is necrosis, and one is apoptosis, which is the programmed cell death. I have given you this figure so that you can refer to it later, that what is the difference between apoptosis and necrosis. Necrosis is basically sudden death which happen because of any wound. Uh, when there is a wound on the body, it happens whenever there is a damage on the body. So cell die directly, there is a sudden death. But in apoptosis, we have something wrong with the cell that is being sensed by the cell, and that leads to a programmed degradation of the cell, because that cell is not right for the body. So that is a programmed cell death, and this is what I have given you so that you can refer to it later. Coming to the next question, which is again, you have to match the drug with their respective mode of action. So we again have paclitaxel. Can anyone tell me now what is the right mechanism of action for paclitaxel? How it interfere in the body? Okay, it, you're saying it's 3. Oh, it's 6. Okay. Just try to remember what I have told you, that it leads to whether, because you have two options. One is inhibit microtubule depolymerization, and one is it inhibit microtubule polymerization. Yeah, it actually depolymerization of it leads to inhibition of depolarization of microtubule. So for P, we have two, which is, I have given you that, if you could see here, that Taxol, which is paclitaxel, it basically leads to depolymerization of microtubules. So for P, we have two. Okay, so then next is colchicine. Colchicine actually leads to what? Colchicine basically leads to microtubule polymerization. So for colchicine we have microtubule. It actually inhibits microtubule polymerization. So what it does, it basically not uh, able to form. It will not let alpha beta polymer binds, so that we can have our microtubule intact. So colchicine have exactly a different mechanism, which actually basically inhibits direct polymerization of microtubule, and Taxol inhibit microtubule depolymerization. There is a constant polymerization and depolymerization happen of microtubule, which leads to cell do various activity, and it is very important. So one is actually leading to uh, one is inhibiting polymerization, another one is inhibiting depolymerization. Both affect our cellular activity and can lead to cell death. And these drugs are used in various diseases. Okay, then we have colchicine, which inhibit polymerization. As I have mentioned, there are different other pathways which is affected by colchicine. You can refer to this particular image for that. Next is Etoposide is a photopolytoxin derivative. It is also used for some cancer. um and it is also used as a cancer drug in some cases. So what it does, it inhibit DNA synthesis. And how it inhibit DNA synthesis? It bind to topoisomerase 2. Can anyone tell me what is the role of topoisomerase 2? Any of you? Just turn on your mic and answer that.
Nicking, I think, I guess, nicking is done by topoisomerase 2.
Yeah, so it leads to the cut of double stranded DNA. So it removes the supercoiled structure of it. It make the DNA. It take it put a cut in a DNA supercoiled structure, which lead to relaxed DNA, and that is very important if you need to do DNA replication. So if there is a inhibition of topoisomerase 2, it will lead to there will be no DNA replication, and then there will be no mitotic cycle. There will be no cell division, and it will lead to cell death. So for etoposide, there is inhibition of DNA replication. Fine. Coming to the last one, which is again MTX. We have already talked about it, that MTX leads to it inhibit DHFA, and it actually halt our DNA synthesis. So if we go back and check the answer, so for Taxol we have 2 P2. So the answer is P2. Then colchicine we have Q6. Answer is B basically. Etoposide we have inhibition of DNA replication, which is 3. And MTX we have inhibition of dehydroxyfolate reductase, which leads to halt of DNA synthesis, and that directly lead to cell death. So this is how you're going to match it. Even if you know one or two option, you can still get the answer, because that's how you can actually eliminate your wrong options. Okay, so coming back to the next question, we just have 10 minutes left, and I'll be sharing this PPT with you. I'm giving you one more important aspect. I'm telling you one more thing which is very important for GATE exam. You need to remember various commonly used drugs and their mode of action in terms of which particular organelle or which particular enzyme they are binding to. I have given an overview. Mostly asked question are from these particular bunch of drugs only. Just refer to these drugs and look for what all. For example, you will find few questions related to vincristine and cisplatin and Taxol, though we have already done. So these are the drugs which are commonly asked in GATE exam. So I've given this figure for your reference only. Coming to the next question, in a typical mitotic cell division cycle in eukaryote, M phase occur immediately after which particular phase? I think most of you can answer this question. Can anyone answer this question for me? सो हमारा माइटोटिक फेज कौन से फेज के जस्ट बाद आता है? Okay, I'll make you remember the organization. So it is G1S G2M. Yeah, G2 is the answer. So always remember G1 से G2 में. ये एक कोड की तरह याद कर लो. G1 से G1S G2 में. G2 से M. So G1 say it goes to DNA replication, which is synthesis phase, and then it goes to G2 phase, and immediately after G2 phase we have our M phase. So the answer for this question is D. And I have given you the mitosis and meiosis and how the haploidy and diploidy changes in mitosis and meiosis. So that this is for your reference only. Next question is which one of the following statement regarding G protein is incorrect? And I'm sure now you can answer at least few things about G protein. First thing is GTP is bound to G protein in a resting state. Is it true? Quickly answer it. We just have very few minutes left. Okay, so first statement is right. Generally, when G protein is inactive, it is bound to GDP. In resting stage, it is bound to GDP. Once we get an hormone, there is a phosphorylation of GDP molecule, which leads to removal of G protein, And once the G protein is unbound, it get activated and leads to further process. So A statement is correct. Second is GTP bound alpha subunit cannot reassemble by beta gamma dimer. It is true, once GTP happen, alpha subunit get detached from the beta and gamma dimer, and it leads to activation of adenyl cyclase. So yes, when alpha subunit is bound to GTP, which is a phosphorylated state, it actually dephosphorylated state. It actually uh, cannot reassemble to beta gamma dimer until unless there is GTP. Okay, so once the dephosphorylation happen, once the phosphorylation happen, it get activated. When dephosphorylation happen, it get inactivated. So if it is bound to GTP, it cannot reassemble to beta gamma dimer. This statement is also correct. So B is also correct. Then we have all G protein are trimeric. Do you think all the G protein are trimeric in nature? They always have three subunit, or G protein might have some less number or more number of subunit as well? So the third statement is basically wrong, because G protein are not always trimeric. They always not have three subunit. There is few G protein which are monomeric in nature. They just have one subunit. Next statement is activation of G protein may result in activation or inhibition of target enzyme. Yes, because this is how the signaling happens. Once the G protein activated, it either leads to activation of certain enzyme or lead to deactivation of certain enzyme. So inhibition and activation both happen. This is the detailed version of how G protein signaling happen. Please refer to this. If you could see that there is a trimeric form, it bounds to GTP, GTP get attached to, okay, GTP, once it bounds to GTP, alpha subunit get attached to adenyl cyclase, it lead to formation of CAMP. Once CAMP is there, there is an activation of various protein. And if it's come inside, it also leads to inactivation of certain protein. So here, the only option which is wrong is A, all G proteins are trimeric. No, because there are monomeric G protein as well, which is RAS. And you might have read about RAS as well. There is this particular, if you're going to use this PPT, there is some type of receptor which I have talked about, which is important for your GATE exam. Please refer to this PPT once you are preparing for once you are revising cell biology. Okay, I have also given what all family G protein family members are there, and how they get, what kind of function they do, and how they get activated, to which particular subunit they bound and get activated. So all the details are given for a G protein. Please refer to these details. This will definitely help you do some of the questions which are from this particular area. Uh, so there are some protein which leads to activation of all. One trick I want to tell you, that is that there is GS and there is GI. Always remember I. For I, it is kind of inhibition. So GI leads to inhibition of adenyl cyclase, whereas GS leads to activation of adenyl cyclase. So always remember, this is a very tricky question. It has been asked in many papers like DBT as well as in GATE, that whether GI is responsible for activation of adenyl cyclase or GS. So these are some tricky things that you have to remember, so that you cannot, because especially जो मैचिंग वाले क्वेश्चन होते हैं where you have to match the two column, वहाँ पर ऑप्शंस बहुत कंफ्यूजिंग होता है. तो you have to remember this thing. GI मतलब inhibition. So always remember, GI will always lead to inhibition of adenyl cyclase, whereas GS stimulatory, which leads to activation of adenyl cyclase. So just try to remember these things. Next question is secretory protein synthesized in ER associated ribosome transverse through mitochondria, peroxisome, Golgi apparatus, or nucleus. So quickly, I'm telling you this thing. I'm not asking whether you know the answer or not, because security, whenever we talk about proteins, translocation or protein folding and other thing, it is always ER and Golgi which come together to do all these things. So whenever we talk about a proteins which are secretory proteins are the proteins that are going to go out of the cell. Fine, they will release out of the cell, and for that they, and once they have, for releasing themselves out of the cell, they need a transportation, and transportation assembly is provided by Golgi apparatus. So whenever there is a secretory protein, there is Golgi apparatus which is involved. And when we talk about non-secretory proteins, which going to remain there inside the cell only, it's mostly nucleus and endoplasmic reticulum that are doing their job. But whenever we talk about secretory protein or plasma membrane protein or outer membrane protein, uh, or receptors and all, they're going to be ER and Golgi working together so that they can reach to their respective places. Okay, so whenever there is a transport required, ER Golgi will work together. When there is no transport required, then ER is working with nucleus. ER is getting information from nucleus and releasing that protein into the cytoplasm. So non-secretory protein, just endoplasmic reticulum, and secretory protein, definitely we need to have Golgi, we need to have transportation of those particular protein to be happen. So the answer for this is Golgi apparatus. Next question is, in eukaryote, cytokinesis is inhibited by. So this drug is very important, and this drug is used in many essays, many research purpose, because it leads to inhibition of cytokinesis, and once the cytokinesis is inhibited, there will be no cell division. So cytochalasin B is used as a cytokinesis inhibitor, and it is being used in many area of research. Fine. So I'm directly giving you the answer here. So cytochalasin B जो होता है वो क्या करता है? Basically, it leads to inhibition of polymerization. So once the way same like colchicine, sometimes colchicine is also used, but colchicine do not lead to cytokinesis. So if you could read about it, cytokinesis or colchicine का थोड़ा थोड़ा difference, so you'll find there is a difference. I'm not getting into detail because I have very less time and I want to cover maximum this thing. But if you read here that it leads to inhibition of polymerization without affecting depolymerization. So सिर्फ polymerization effect होता है. Polymerization का मतलब है कि अब orientation होगा ही नहीं, microtubules बनेंगे ही नहीं, and if there is no microtubule formation happening at the division, तो वो जो हमारा फरो बनता है ना, जो डिवाइड करता है, वो होगा ही नहीं. एंड there is no division, if there is no assembly of microtubule there will be no cell division. So cytochalasin D is the answer for this particular question. If you could read here, both cytochalasin B and D are responsible for cytokinesis. They both act similarly. Next question is where you have one group of proteins or enzyme, and you have to talk about they lead to what particular cellular processes. So number one is P53. Can anyone answer this thing quickly? Because P53 is a very known protein, and it leads to what? Answer. I'm looking for some answers here. Yeah, it leads, no, it's, I mean, you are giving me answer directly. I'm asking what P53 does. P53 leads to apoptosis. It is involved in programmed cell death. So for P, it is 2. So for P, it is 2. Next is lysozyme. Lysozyme. What lysozyme does? Can anyone tell me? Okay, lysozymes are the enzymes that are present in saliva. So what it leads to? These enzymes basically leads to hydrolysis of polysaccharides. So whenever we have some particular whenever we eat, yeah, so it's 3, right? It leads to degradation of polysaccharides. Then we have tubulins. Tubulins, as I told you, that tubulins are the ones that leads to movement of things. माइक्रोट्यूबुलेंस से ही हमारा डिवीजन होता है पूरा. तो माइक्रोट्यूबुलेंस का क्या होगा? Yes, chromosome segregation, because microtubulins leads to the division of the movement of sister chromatid towards their respective pole. And then we have histones. Histones leads to DNA packaging. So P53 2, yeah. So P53, yeah, S का 1, P2 हो गया, Q3 हो गया, R4 हो गया. So I think it's D. if I'm not wrong, histone 1 हो गया. So I think it's D. I have given all the detail that P53 is involved in cell cycle, cell death. Lysozymes are the one that leads to degradation of polysaccharide, and it also protect our body from the bacterial attack because it leads to degradation of bacterial cell wall as well. Then we have tubulin. Tubulin leads to the movement of sister chromatid towards their respective pole, and then we have histone, which is responsible for the DNA packaging inside inside our into the chromosome, because there are large, huge amount of DNA which is packed around the histone protein, and that's how our chromosome look. Next question is, the genome is deployed at the end of which of the phase of human mitotic cell? So whenever there is a division, there is a time when our cell is deployed, which is a normal form of our cell, and there is a stage where we have uh, where our cell is not in a diploid form, is mostly in the tetraploid form. It is 4n in nature during the division. So can anyone answer this thing? Okay, fine, I'll take you through this thing. So what happened when we have our G1 phase, which is normal assembly of all the protein getting ready for the cell division, we have our diploid D maintained, because that's how we have our chromosome in a form of 2n, right? You remember two set of pair of 23 chromosomes, so that is diploid. When there is a DNA replication, that means that DNA is getting replicated. That means अब क्या होगा, 2n से 4n हो जाएगा. तो यहाँ पे क्या हो गया, 4n. तो हम at the end of the G1 we have diploid. Remember G1 पे diploid है. अब यहाँ पे 4n हो गया, तो यहाँ पे तो डिप्लॉयड नहीं है, तो मतलब एस फेज तो इसका आंसर नहीं होगा. अब G2 पे, बिकॉज अभी डिविजन नहीं हुआ है, तो G2 पे भी ये 4n ही होगा. But once there is a division, once the mitotic stage happened, there is again diploidy maintained. So the answer going to be G1 and M, because at S stage we have 4 and at G2 stage we have 4. So all the option have S or G2, so they are eliminated. We have B as an answer. So the answer, sorry, what the hell is this? No, it's going to be B only. I think I have written wrong here. The answer is B. That is G1 and M. At the end of G1, we have diploidy maintained, because they're going to be S phase where we have 4n. And at the end of mitotic division, when there is two sister daughter cells are divided, they also have diploidy maintained. You can refer to this particular because we are referring to mitotic, so you have to refer this. When we are talking about meiosis, you have to refer this. So at the end of mitotic division, we have diploidy maintained again. So here I think I have written wrong. Please correct it. This is B. Okay. Next question is, I'm taking 5 more minutes. I hope that is fine with you guys. And next time we'll start exactly at 3 p.m. So I want you to join exactly, 2 or 3 minutes before 3 p.m. Next question is, cell type A secretes molecule X into a cell कल्चर मीडियम. तो एक सेल है A जो कि एक मॉलिक्यूल को सिक्रीट कर रहा है X culture media में, and then there is another cell B in the same culture, that means in the near vicinity there is a cell type B which is responding to this particular molecule which is being secreted by A. तो वो A का जो molecule secrete हुआ है उस पे action किस पे हो रहा है, B पे, और B क्या है, उसी particular culture में present है, same culture में present है, मतलब near by present है. तो which one of the following mode of signaling represent the interaction between A and B? ऑटोक्राइन, एज आई कैन टेल यू, ऑटोक्राइन क्या होता है कि अगर मान लो एक ही सेल है, उसी से कोई चीज रिलीज हुई और उसी के ऊपर एक्ट हुई, द सेल टारगेट इटसेल्फ, सेल रिलीज अ मॉलिक्यूल एंड इट has an effect on the same molecule. That is autocrine on its own. Auto means on its own. So autocrine is a cell target the cell. Second one is signaling across gap junction that happen in the cells that are next to each other, जिसे हम juxtacrine भी बोलते हैं, which is next to each other. Then we have paracrine. Paracrine में क्या होता है कि एक सेल ने रिलीज किया, पास में ही एक दूसरा सेल है, उसने उसको ले लिया. तो that happens when the cells are in nearby vicinity. So A ने रिलीज किया X molecule, nearby B था, B ने ले लिया. So we have the answer, that is paracrine. But in case of endocrine, when one cell release a molecule, it goes to the bloodstream, it reach to a far uh, placed cell, and there it shows its action. That is endocrine, that happen through bloodstream. So for example, brain ने कुछ रिलीज किया, bloodstream में गया, और stomach को कहा कि उसे भूख लगी है, for example, like that. I'm just trying to be funny here, sorry. But yeah, it's like that. So endocrine is basically when a cell from the different tissue release a molecule, it comes to a bloodstream, it reach to a distant tissue or organ, and their cell there take that particular molecule as in it has an effect of, those cell has an effect of that particular molecule, which are placed far apart from the releasing cell. So that is endocrine. So here we have a question where they are saying कि वो nearby में ही है cell, because it's in the same cell culture medium. That means a nearby cell. That means the answer is paracrine. So the answer is C here. Okay, last question for this for today is this, that so we have organelles, and we have to talk about what kind of function they do. So first one is lysosome. Lysosome leads to what? What do lysosome do? Lysosome, can anyone answer that? अभी हमने बात की लाइसोजाइम, लाइसोसोम में प्रेजेंट होता है वो. Okay, yeah, one. It digests the foreign substances. So lysosome contain lysozymes, and these they contain other enzymes as well, and it leads to the digestion of foreign substances. So P is one. Then we have smooth ER. What does smooth ER do in the cell? So smooth ER is basically there is no ribosome on smooth ER. Smooth ER is without ribosome. So as such, there will be nothing but, yeah, it leads to lipid synthesis, because smooth ER is responsible for lipid and carbohydrate synthesis only. It does not involve in protein synthesis. Protein synthesis happen at RER, which is राइबोसोम कंटेनिंग ER, जिसे हम रफ ER भी बोलते हैं. ठीक है, तो स्मूथ ER का ये हो गया. देन वी हैव गोल्जी apparatus. गोल्जी apparatus लीड्स टू, गोल्जी बॉडी इज इन्वॉल्व इन ट्रैफिकिंग. सो उसी से पता चलता है कि वो प्रोटीन टारगेटिंग करता होगा, मतलब वो प्रोटीन को एक पर्टिकुलर जगह पे लेकर जाता होगा. तो R का हो गया 2. And then we have nucleolus. So nucleolus leads to uh, rRNA synthesis. It does not involve in any other RNA synthesis, but only rRNA synthesis. So if you could see, nucleolus is present inside the nucleus. It has granular component which actually assemble rRNA, and it leads to ribosomal assembly. Ribosomal assembly. Fine. So it create that assembly which leads to uh, activation and deactivation of certain genes. Okay, so that is there in nucleolus. So if you go by the option, the answer will be A, that P lead to P, that is lysosome, digestion of a foreign substances. Smooth ER leads to lipid synthesis, and Golgi apparatus actually leads to protein targeting, because it involves in protein trafficking. And nucleolus, it leads to rRNA synthesis. It is not nucleolus. There is no direct protein synthesis here. Protein synthesis happen outside the nucleus. So that is why fourth option is eliminated, and the answer is A. So there are a few more questions. I think RAS wala now you can do. It is a monomeric this thing. And last one is mitochondrial. These are the subcellular organelles, and you have to relate to its something which is given in group 2. So mitochondria, as you remember, there are 4 options here: single membrane enclosed, double membrane enclosed, maternal inheritance. So maternal, the moment you have maternal inheritance, maternal inheritance is always associated with mitochondria. So P3. So you can eliminate rest 2 option. Now you have COD left. Then we have chloroplast. Chloroplast, have you read about endosymbiotic origin? So what exactly symbiotic origin is, that things are originated from our precursors. So mitochondria and chloroplast both have symbiotic origin. They are originated from our precursor cells like ancestral eukaryotic cell. For example, माइटोकॉन्ड्रिया को बोलते हैं कि वो पर्पल नॉन सल्फर बैक्टीरिया से आया है, और क्लोरोप्लास्ट को बोलते हैं it is evolved as a from the cyanobacteria. So being a eukaryotic cell, they are evolved from the prokaryotic cells. So that is called endosymbiosis. That is a endosymbiotic theory. And that's why if we are giving mitochondria as a maternal inheritance, endosymbiotic both can go to mitochondria as well as chloroplast, but here we have mitochondria for maternal, so we are giving Q to 4, which leads to the option, I think, A, Q to 4, because there is no, sorry, Q to 4 C, because A and B are eliminated because mitochondria is associated with maternal inheritance. For Q, chloroplast, we have endosymbiotic origin. So Q4, here we have Q1, which is single membrane. No, chloroplast is not a single membrane organelle. It is a double membrane organelle. It has inner as well as outer membrane. So D option गया. So अगर C में देखें आगे R2, न्यूक्लियस. न्यूक्लियस हमारा क्या होता है? न्यूक्लियस हमारा डबल मेम्ब्रेन इनक्लोज बॉडी होता है, because it also have इनर एंड दी आउटर मेम्ब्रेन. न्यूक्लियर की इनर मेम्ब्रेन भी होती है, आउटर मेम्ब्रेन भी होती है. सो नेक्स्ट वी हैव एंडोप्लाज्मिक रेटिकुलम. यस, एंडोप्लाज्मिक रेटिकुलम इज सिंगल मेम्ब्रेन ऑर्गेनेली. सो दैट्स हाउ वी कम टू द आंसर. अगर आपको दो ऑप्शन भी पता होंगे ना इसके, आप ये क्वेश्चन कर सकते हो. तो दैट्स हाउ यू रिलेट टू द ऑप्शन. सो इसका आंसर है, ये थोड़ी डिटेल्स है रिगार्डिंग दैट. देन देयर इज अ सिंगल मेम्ब्रेन ऑर्गेनेली, लाइसोसोम, एंडोप्लाज्मिक रेटिकुलम एंड गोल्गी apparatus. Chloroplast, nucleus, and mitochondria, they are double membrane. They have outer as well as inner membrane. So that's how you can come to the answer, which is C here. That's it for the today. I hope you are, I know we have to rush it very quickly with all the questions, but these PPTs and the videos related to this particular session will be shared to you, so you can refer. These are the very important aspect of the GATE examination. If you cover these particular subtopics, you will be able to solve cell biology at least. So that's how that's the ending of my session. If you have any question, you can come. If you have any suggestion ki next class mein how I should teach you, what all things I should give you, let me know either in comment box, or you can switch on your mic and tell me right now. This is the first class. I want you to stay connected through the entire session. This will definitely help you crack your examination, because that's how we're going to relate to कि हमने आज सेल बायोलॉजी किया, कल हमने मॉलिक्यूलर बायोलॉजी किया, तो हम कैसे प्रोग्रेस करेंगे. एंड देन वी हैव बायोप्रोसेस इंजीनियरिंग एंड वो वाला एस्पेक्ट भी. सो राइट नाउ I am starting with few very easy subject. I think cell biology, molecular biology are the subject that are very scoring one, because you will not make mistake in those subjects. Then we'll move to other biotechnology subject as well. So for that, stay connected. I'll be taking class every Saturday 3 to 4. And if you have your friends, your anyone who wants to prepare for GATE examination, you can ask them to join this portal. This is free for everyone. You have different courses running together, different subjects running together, and according to your availability, you can take days. So for Saturday, just say for example, I'm taking your classes, and if you have any suggestion that how I should proceed, because these I have covered maximum cell biology question for last 10 years. 2011 से 2020 तक के तो हमने cover कर लिए हैं. तो अगर आप इन सबको भी गो थ्रू करोगे ना, आपको समझ आ जाएगा कि कौन कौन से क्वेश्चन पूछे जा सकते हैं, और उन्हीं के आसपास आएगा. जैसे फॉर एग्जांपल ड्रग्स का, दे आर रिपीटिंग द ड्रग वाला क्वेश्चन. कहीं पे वो उनका मोड ऑफ एक्शन पूछ रहे हैं, कहीं पे उनका किस पर्टिकुलर एंजाइम से बाइंड कर रहे हैं वो पूछ रहे हैं. सो दैट्स हाउ यू गोइंग टू रिलेट टू इट. आपने एक बार उसको पढ़ लिया, आपको वो हमेशा याद रहेगा. एक बार आपने उसको पूरा अच्छे से पढ़ लिया कि एक पर्टिकुलर ड्रग का क्या एस्पेक्ट है. So now I'm ending my class. If you have any question, please, I'll give you 2-3 minutes. You can talk, you can switch on your mic and ask me. And if you want कि next class से थोड़ा इस तरीके से मैं आपको पढ़ाऊँ, जहाँ पे मैं concepts भी बताऊँ आपको, या Shansh, if you just switch on your mic and say whatever you want to say.
Yes, ma'am. So today's session was quite nice, ma'am, and thank you for that. But next time when you teach, when you come for this session, Ma'am, please make sure you elaborate the some topics like, say, for example, protein shorting and
Okay.
protein packaging. So,
Okay.
before jumping on to the direct question,
Mhm.
you just briefly describe the how the things happens, and
Mhm.
you are getting
Yeah, yeah. Okay, fine. If you
बताने से
want
पहले
to,
एक तो yes ma'am.
yeah,
So
you are at,
different so difficult topics को एक बार questions बताने से पहले आप थोड़े उसके theoretical portion को आप एक briefly describe कर दो मैम. That's
ठीक है.
okay. And
ठीक है, I think I'll try to cover the tough topics because we just have 1 hour. Okay, fine, I'll do this thing that I'll make some extra additional, notes in that particular question only so that you can understand what exactly clonality trafficking or cell sorting or DNA packaging is. I'll try to cover that, but okay, fine. I'll keep that in mind. I'll try to cover concepts as well. I think that is important. Anyone else want to share anything?
and ma'am, where we are supposed to get the PPT of this
You
session
so
on
you will be shared resources on the NPTEL website only. जहाँ से आपको लिंक मिला था वहीं पे ही आपको बता देंगे वो. आप वहाँ से देख सकते हो. और other resources में, as I have mentioned, in next class what I will do, I will point out some very important topics that you have to read, and I'll make a list of that. Even if I'm not giving you a resources, you can Google those
yeah, yeah
subtopics
ma'am,
and you can read about those topics in
no
detail.
problem ma'am, thank
That
you
will
very
help you a
much.
lot. Okay. And I would ask you to convey about this particular portal to as many student as possible, all your friends, all your colleagues, college mates,
Yeah, sure.
so that they can also access this particular, free service for you guys. So that will help a lot of people. So do that and stay connected for the next class. I want you to come. In the next class, I'll be taking molecular biology. I would ask you to read more about molecular biology, try to revise the topic so that whenever we do the question related to any of the topic, you will be able to do that question without knowing the answer. I mean, if I'm going to, if you're going to give me the answer of that particular question, that will be more, uh, you know, uh, that would be more good. I mean, even I'll think, yes, you guys are preparing for that particular subject. So in next class, I'll be taking molecular biology. Molecular biology is a very vast subject. I'll try to cover as much as possible through questions only. But at the same time, I want you to read the subject, at least read the highlight of that subject so that you can continue with that. So you can do the question at least, few question at least. Fine. So I'm ending my session. You guys can leave the meeting. And thank you for joining and all the best for your exam.